Hello, this is Dr. Patrick Hu, CEO of Moffitt Cancer Center and president of SITC. I'm just so happy today to have my good friend, Dr. Ron Levy from Stanford University as our guest. Dr. Levy has really been an icon in the field of tumor immunology. He was one of the first people to treat cancers with antibodies. He did some of the very early cancer vaccine studies and is continuing to lead the field in the immunotherapy of lymphoma. So thank you, Ron, for being with us today.
Thank you for having me. It's really a pleasure to be here.
Wonderful. Let's start from the beginning. You got your bachelor's degree at Harvard, then you went to Stanford University for your medical degree. Tell us what got you interested in tumor immunology.
Well, I have to say that it was something that had to do with my mentors, my teachers, my peers, but also in those days, Stanford had an experimental program of medical school where you were obliged to stay for 5 years rather than the typical 4 years. And they spaced out the time over the 5 years of assignments, so you could save it up and do various things, including research projects, travel, get other degrees at the same time. So I was fortunate enough to be able to associate myself with one of my professors during that time, Henry Kaplan and then Saul Rosenberg. And these were giants in the field of cancer, and particularly focused on lymphoma, but also on the immune system. And there was a visitor who was a sabbatical visitor, professor from Israel, from the Weizmann Institute, named Michael Feldman, who visited Henry Kaplan. We had a journal club at Henry Kaplan's home for medical students to come in the evenings and discuss publications and papers that are written in the field. And cancer immunology really seemed like a great idea. I mean, it seemed like the immune system and cancer had something to do with each other at the time. Little did we know what it would eventually become. But it was fascinating to me how the immune system of the body and how the biology of cancer seemed to be related at that time. There should be ways to take advantage of the immune system to diagnose and treat cancer better. So that's where it all started in my medical school days with Henry Kaplan and Michael Feldman.
Well, that's exciting. Well, you really did work with giants. I remember Michael Feldman from the Weizmann. He was an amazing guy. I remember Lee Eisenbach, I think, worked with him there at the Weizmann. So tell us, you really pioneered some of those early studies with antibody therapeutics as well as vaccines and in lymphoma therapies. Tell us how that came about.
Well, I remember the day as a medical student when I was in the clinic and they were discussing a case of chronic lymphocytic leukemia that had been seen that day. And they were talking about the restriction of the light chain of the variable region of immunoglobulin as a way of diagnosing the clonality of chronic lymphocytic leukemia. And because I had studied immunology and knew something about antibodies at the time, it really became apparent to me that every patient would have a unique clone that could be identified by the immunoglobulin that the clone made. And each one would be different from each other, but different also from every normal cell in the body. And that idea of what became known as the idiotype, the unique portion of every patient's lymphoma or leukemia, seemed like an obvious target to me to go after. Let the immune system go after. And so that idea really stuck with me to the present day, actually. We're talking 50 years of time here, where that seems like a really good target, customized for the patient, but very specific for the tumor itself. And the question became how to target something like that. When I was at the Weizmann Institute for my medical student rotation, and then later as a postdoc back at the Weizmann Institute, I came in touch with a postdoc by the name of Norman Kleinman, a basic immunologist, who was making monoclonal antibodies by cloning cells from one animal to another. So he would immunize an animal, take the spleen cells from the immunized animal, and then transfer them to his identical twin animal, a syngeneic animal, in limiting dilution. intravenously. And then the cells would land in the spleen of the recipient animal, and he would chop up the spleen into little pieces and culture those pieces. And those pieces might have gotten none or one of the donor cells from the donor vaccinated animal. And those cultures that he was setting up, I remember asking him, what's the value in doing monoclonal antibodies? Why do you do this? And he said, well, we want to find out how the immune system works and how diverse it can be and how to trigger it better. And it was cloning the cells in vivo and then capturing them in little spleen fragments that were cultured in vitro. So when I came back to Stanford, I set up that system for making antibodies against leukemia and lymphoma cells, thinking about the target that I had in mind, the unique target, the idiotype target. And the thing about those cultures is that they produce really high quality monoclonal antibodies for very limited durations of time, a couple of weeks, and then they would die out. But you could harvest the supernatants from those little cultures and do a lot of analysis with the antibodies that were monoclonal, single products of single clones, expanded in those cultures for a few weeks. And then they would die out. And just about that time, Kohler and Milstein published their famous paper on how to immortalize the antibody producing cells by making hybridomas. And Len Herzenberg at the time was very interested in doing that as well. And we were able to get the cell line that Kohler and Milstein had used to make their hybridomas and to transform our work from making limited duration, limited quantities of monoclonal antibodies to making antibodies that would live forever and be available forever. We still have some of those original hybridomas that we made in 1975 or 1976, still growing or still living and resurrectable from the freezer and making the same antibody that they made 40 or 50 years ago, which is incredible to think about, that to capture the cells that make the antibodies and have those antibodies, if they're really antibodies you like, to be able to work with them forever and never have them change. That's really, and that turned out to be the basis for so many immunotherapy ideas that came subsequent to that time. So that's a long-winded answer. It kind of takes you through a process of realizing opportunity and taking advantage of discoveries that other people make and applying them to a problem that seemed like a tractable problem, the problem targeting cancer.
And you still see patients in the clinic, right?
Every day when I see patients in the clinic and use rituximab or other antibodies that we have, or CAR T cells that we now have available due to other people's discoveries, all derived from monoclonal antibodies one way or the other. Checkpoint antibodies, CAR T cells, direct killing antibodies like rituximab and derivatives and other versions of that idea. When I see patients in the clinic, I just have this amazing gratitude for what's happened over these years and what we have available to us now that we didn't have when I began my career in the clinic.
Yeah, it must be incredible because you were there from the start of the development of monoclonal antibodies, and now monoclonal antibodies are just something people take for granted and are in so many aspects of therapies, whether direct, conjugated to radio, nucleotides, conjugated to chemotherapies, bispecifics, so many different versions, and also on the CAR Ts. So it must be really gratifying that you were there from the start.
Well, I actually worked on the problem of making an antibody drug conjugate when I was in my postdoc in Israel in 1973. And it seemed like a good idea if antibodies could target to the tumor and you could couple a drug to it. So I actually used some chemistry that was invented at the Weizmann Institute to link adriamycin, the drug adriamycin, to antibodies and to deliver them in the body in mice to tumor cells in mice. And it took a long time before antibody drug conjugates became a useful form of therapy in the clinic, but that idea seemed like opportune at the time. It's interesting how ideas can occur, but applications and development drugs in the clinic that we use in patients take a long time to happen, even with a great idea.
Tell us about the development of Rituxan, which is one of the mainstays of therapy for lymphoma right now.
Yeah, Rituxan was an interesting development. We, in the lab, had made antibodies for individual patients against their idiotypes that I described before, and actually used them on patients successfully to treat their lymphomas. And there was a student who was working on it at the time, whose father was a business developer, and he asked us, "Is this a treatment that you would want if you had this disease?" And we said yes. He said, "Then we have to make a company." And so the company that was made on that idea was called IDEC for idiotype. And IDEC made antibodies custom made for patients. And at a certain point, the management of the company decided that that was not a good business, customized therapies. And so they wanted an antibody that would work for many different patients. And about that time, investigators in Boston, Lee Nadler and others, had developed antibodies to B cells being the cell of origin of most of the human lymphomas and leukemias. And the company actually made an antibody that became eventually the drug Rituxan. And due to a lot of really interesting, lucky accidents, accidents of the target, accidents of the production of the antibody, accidents of the effectiveness in the clinic and the lack of side effects, none of which could be predicted, it became the drug Rituxan that we use every day in the clinic, even to the current day. And I actually didn't think it was a very good idea to go after B cells as a generic target. It wasn't as customized for sure, but it also wasn't as specific for the tumor as the target I was more interested in, which is the idiotype at the time. And we didn't know it would be safe. We didn't know that the depleting B cells, normal B cells of the immune system would be a safe thing to do. We thought it might cause immune deficiency. And to a certain degree it does, but it's tolerable, it turns out, and that was unpredicted. And we did the original clinical trial with the antibody that became known as rituximab. And about half the patients responded to that therapy, off the shelf, ready to go, one size fits all, a product of cloning the genes for the antibody and finding a good producing cell, making enough quantity to do the clinical trial, and proving to the FDA that it actually worked and didn't have many side effects. And it's the no side effects that really made it a viable drug. The lack of some of the predicted side effects that didn't happen, and the efficacy as opposed to the safety of the drug. We had other drugs, chemotherapy, that had the same efficacy, but not the same safety. It really was the safety that convinced the FDA to approve the first monoclonal antibody for treating human cancer.
Wow, yeah, it's such a critical role in that really seminal work. You train a lot of fellows too. When they come to your clinic at Stanford and they just give R-CHOP or Rituxan-based therapies, do they have any clue of the history and your role in that?
Very little, actually. Someone once said to me that clinicians need to feel like they own the therapy before they'll adopt it and use it. And I think this is a mark of success in the field, in the marketplace, and in the field, that people don't need to know the history, but they need to feel like they own the drug, own it in an intellectual sense, in a therapeutic sense, not necessarily in a financial sense, but they need to think of it as foundational and something that they use as a tool that they rely on. And that became Rituxan's a legacy, actually, in the field.
Wow. Well, you have been recognized for it. In 2009, you got the King Faisal Award. You went to Saudi Arabia. That's known as the Arab Nobel Prize. What was that like? That
was an exceedingly interesting experience. When the dean nominated me for that prize, I didn't think that they would take my candidacy seriously. having so many connections to Israel and the Weizmann Institute for my postdoc training and medical student rotations, it was on my CV, of course, and everybody knew about it. And I didn't think that Saudi Arabia, a prize based in Saudi Arabia, would be something that they would be interested in me. And then when they contacted me and said that they had chosen me for this prize, I went to their website and I saw my picture on their website and my CV, and it was really interesting to see that. In the course of going there, of course, we could bring our families, and I have 3 daughters, and my wife, who's Shoshana, who's a collaborator with me all these years, and scientific collaborator, as well as my spouse, and so I was taking 4 women to Saudi Arabia, educated women, and they became role models on that trip for other women in Saudi Arabia who we met, and they were interviewed on television in Saudi Arabia television, and we were ambassadors actually for science and for the potential of everyone in science and women as well. And visiting, being in Saudi Arabia as a award winner, there was the award ceremony and the chance to meet the king and to give a speech and to meet scientific colleagues. But they were really interested, the Saudis were really interested in what we knew about them and what we thought about them. They introduced us to a lot of colleagues and scientific colleagues and medical students and different parts of their country, and did a feast in the desert under tents with open fire pits, cooking food and letting us ride on camels, and it was just an amazing experience, culturally and socially as well as scientifically, and very surprising to me that they would consider me with my background for that award.
Well, just so well deserved. Now you still have a very active lab. You're putting out incredible papers in the literature right now. Tell us what you're most excited about.
Well, in the last, after Rituxan happened and other things that we tried worked not as well as we'd hoped, a postdoc came to my lab with an idea of making dendritic cells and injecting them directly into tumors, where they would load with antigens directly in situ in the tumor, and then trigger the immune response on the T cells locally at the tumor site. And he had done this experiment before coming to my lab, and he brought it to my lab. His name was Winru Song, and he convinced me to try it in our lymphoma systems, and it worked. And then I was introduced to the idea of toll-like receptors as molecules that could do similar things. So in the last decade, we've been developing the idea of locally injecting immune stimulants like toll-like receptor ligands directly into tumors, triggering an immune response that goes body-wide and treats tumors at uninjected, untreated sites in the body, so-called abscopal tumor responses. This is an old idea. It was worked on by Irv Bernstein and other people at the NIH many years ago. But now we have new tools to bring to the idea. Immune stimulants, antibodies, triggering the T cells, toll-like receptors, triggering antigen-presenting cells, and the ability to trigger an immune response by local injection. This idea of in situ vaccination, you can apply it to metastatic disease where you have an injectable site in the body, one of the metastases, and look for responses not only in the injected site, but also in the metastases. It's also an idea that can be brought to the front for neoadjuvant applications. And we have a recent publication doing just that in the animal model, where you would inject to the tumor that you know where you're going to resect it surgically, where there already might be metastatic spread that you may or may not know about. And then you can leave behind a trained immune system after the surgery that goes after the metastases. So I think this is an idea that has a future. We've been impressed with our ability to make this work in animals, and we're doing clinical trials in people, particularly in lymphoma, which is my clinical specialty. And we have very interesting abscopal responses occurring in our patients. based on this idea. And the problem becomes what to inject. What are the best molecules? What are the best triggers? How do they work together in combination? So it's an ongoing project, both preclinically and clinically, and then analyzing the specimens that we get from the patients back to the lab to analyze the effects that we're having on the immune system locally and distantly in the body. So this is the project we've been working on most recently.
So it's really to try, instead of finding the antigen and injecting it like the COVID vaccine and other cancer vaccines, it's to use the tumor itself as the vaccination site by injecting immune stimulants, generating the immune cells, and then having them go throughout the body to try to fight the cancer.
Yeah, and the advantage is we don't have to pre-identify the antigen. The antigens are there in the tumors based on somatic mutations, probably, and they might be different in every patient. And the immune system is present locally and can be activated in many, many instances by the right signals to defeat the negative effects of tumor on the immune system, and also directly stimulate the immune system and stimulate antigen presentation by the dying tumor cells, released from the dying tumor cells. So that's the general idea. And now some of the tools that we have to do this with are really interesting. Now with RNA, RNA can be coding for antigens and also be coding for immune stimulatory molecules, and delivering RNA as a way of triggering the immune response is part of the idea that we're collaborating with some really, really good chemists here at Stanford that we've been fortunate enough to collaborate with to deliver RNA by delivery agents, polymers that they've invented that protect the RNA and release it for antigen expression in the tumor microenvironment. So that's the extension of the in situ vaccination idea with new tools. And the thing that I've learned through my career is that the problems of cancer are always the same. The patients are always the same, but the technologies move. And the developers of the technologies may not always know of the problem that we're interested in solving, but they provide new and better tools continually to address the problems that we have in the clinic with patients with cancer. And I think that's going to continue to happen as it's happened in the time that I've been in this field. Seeing these developments starting with hybridomas and cloning genes, making antibodies, making derivatives of antibodies, making cells that have antibodies as their recognition units, and making molecules and delivering molecules with more and more sophisticated technology. It's been exciting, and it's still very exciting. I'm just as excited now as I was when I began in this field so long ago.
That is exciting. There's a lot of progress, and your work has really been critical in moving that progress along. You focused mostly in lymphoma for these in situ vaccinations. Do you think they have a role for breast cancer, colon cancer, some of the epithelial tumors?
Well, our preclinical models, these ideas work just as well in models of solid tumors, all of those types that you just mentioned, as they do in lymphoma. In the clinic, I have my super specialty of lymphoma, and my own patients in my own clinic are my best source of referral for our clinical trials. So as I think many of us know that to be a clinician, you have to be a super specialist in one narrow field. And my focus on lymphoma is because my clinical focus is on lymphoma. But the ideas of tumor immunology go through the spectrum of all cancers. And turn the light back on here. So the ideas of triggering the immune response, either with checkpoint antibodies or direct injection into tumors, these ideas go well beyond hematologic malignancy. And we don't really know which are the best tumors and tumor types to apply these ideas to. But I can tell you in the preclinical models, breast cancer, colon cancer, melanoma, a variety of solid tumors work in this modality.
Throughout your career, if I look at a SITC meeting and I look at the leaders in the field, so many of them came from your group. There's a Ron Levy trainee, there's a Ron Levy trainee. And so tell me, what does mentorship mean to you, and what do you recommend for young people now getting into the field?
Well, I encourage people to learn as much as they can about science, get their degree in science and in medicine, both, and to embark on careers that bridge the gap between scientific discovery and medical applications. I think it's a wonderful field. I think that a lot of people are really great at science, but sometimes don't know the clinical problems that we want to solve. They're the people who take care of the patients and don't know the tools that are available. But the career of combining the two is a fantastic career, and one that sustains us as developments keep happening. And so the trainees that I've trained are all doing this to one degree or another. And as you mentioned, many of them are famous in their own right now. And Aurelian Maribel, who you may know, recently had a sabbatical after being a postdoc here 10 years ago. And when he left, he gave me a what looks like a diagram of a computer chip, which summarizes all our publications and all our co-authors and all the years, all connected by lines, that summarizes a long period of time of trainees and collaborations and outputs, scientific outputs, that summarize a long career in this field. And I just encourage other people who are thinking about this career to embark on it, and to guarantee that there will be opportunities to make discoveries and to make new applications and to cure more people with cancer. That's our goal. That's
great. And you're right. Your trainees really are focused on that, bringing basic science to the clinic and back. They're some of the best translational researchers in the field, based on your role model, you as a role model. Last year also, you received the honor of being one of the first inductees into the Academy of Immuno-Oncology with SITC. Tell us about that and what that means to you.
Well, SITC is an amazing organization. One of our colleagues once said to me, "This field used to be fun when it was just us." Actually, it's not just us anymore. It's everybody's in this field. And SITC represents the frontier of applying the immune system to cancer. And all of the champions of this goal are recognized, and to be among the group of them who are recognized in that way is really an honor for me. And just I think the purpose of the larger purpose of recognition and prizes and these things is to encourage new people to embark on this field and to rejuvenate it so that they can become the honorees someday. And I think that's the real value of doing and having this recognition and doing this recognition of other people, to encourage new people to embark on this pursuit.
Yeah, well, certainly your career certainly has been inspirational to see where you've taken therapies from really having very little for patients to now having a lot for patients, and following your path, we can just continue to drop that death rate against cancer in the coming years. You have lived in California for a long time now, and every time I visit my daughter in California, I think, whoa, the weather is just perfect here. It's I don't know, do you get tired of the good weather? What do you like about California, and what do you generally do for fun outside of work?
Well, my family lives in California, and I grew up here and went to high school in this same town where Stanford is located. So for me, it's home. And during the days when I was making customized antibodies for individual patients, it was my anchor to stay here, because there's no way I could do that for that patient with that antibody somewhere else in the world. I had to be doing it here for that patient with that antibody here. So there was a hook scientifically in my career, and my goal in my career to keep me here. But it's been a terrific place. I mean, like a lot of places, it's grown enormously over the years. And the science that's going on here is exceedingly exciting. Being on a study section, for judging grants at the NIH, taught me that Stanford's not the only place in the world, that there are very exciting things happening and very amazing people all over. And it gave me a more world view of what we're doing rather than a local view. But as you say, the weather is good here. My family is here, and I think all of us develop roots in places where we work and live. And so I've had only one job my whole career, and that's been here at Stanford. Not that I haven't occasionally been tempted to look elsewhere, but I've never found a better place. So what do I like to do? I like to ski. I like to ride bicycles. I ride a bike to work every day. I like to visit family, and they're nearby. So, and we do a lot of traveling for fun together, our family, and that's what we enjoy. And I mentioned my wife Shoshana, who's my scientific collaborator as well, and we run what we call a mom and pop lab here, where our labs are next door to each other, and we share a lot of people and facilities and projects, and that's been a very sustaining part of my career, having a spouse who's also a scientist, where our shop talk at the dinner table was outlawed by our kids very early on, but we always lapsed into it eventually in the course of doing our work together.
Oh, that's great that you guys have been such great partners. I remember talking to the two of you, and you just telling me that through your scientific travels, you guys have been to just about every country in the world, so many places around the globe.
Yes, I mean, we have a lot of professional travel, all of us do in this field, and then we usually find ways to enjoy those places that we go to, wherever they are in the world. So traveling for business and traveling for fun can be combined. As I said to one of our granting agency people, if we're together, we're home, wherever we are in the world. So that's part of our partnership, and scientifically and in every other way.
All right. Have you and Shoshi, you're working together a lot, collaborating a lot together. Have you ever had a scientific disagreement, and how do you resolve that?
We try not to argue in front of people we work with. We save the arguments for other times. And we often have differences of opinion about the best course to take, or the best experiment to do, or how to advise a person who's trying to decide what to do. Yeah, I grew up in a family that ran a small business, and everyone in the family worked in the business. So the idea of working in a family setting professionally is not a new idea for me. And I think the same was true for Shoshana in her family as she grew up. So the idea of having a career connected to each other is not a foreign idea to either one of us. And we managed to make decisions, even though sometimes we disagree.
That's great. I'm trying not to get you in trouble here.
I'm trying not to get into trouble here.
Did you guys meet in the lab?
Actually, we did. We met in Israel in the Weizmann Institute. As I told you, I followed Michael Feldman back to the Weizmann Institute as a medical student. And the day I arrived there, I said to Michael Feldman, "I'm from California. Do they have surfing here in Israel?" And he said, "Oh, yeah, we have surfing. Here's Shoshana. She'll tell you all about it." And that's how we met. At the time, they didn't have surfing in Israel. He made it up. But that's how we met. And we both worked together with one of the postdocs who was there at the Weizmann Institute, the one I mentioned before, Norman Kleinman, who was cloning those B cells in spleen cultures. And we were both encouraged to get together by him. And he took us on excursions with his family to see different parts of the country at the time. And that's where we met.
That's a great story. That's a beautiful country. So have you and Shoshi ever been surfing together in California?
Well, we don't surf. I do windsurfing, but she never picked that up. But we do kayaking. A double kayak is a stress to family life. Talk about trouble making decisions. We were once kayaking in Hawaii at a scientific meeting, kayaking, and the whales were jumping out in the as they do in Hawaii, and I was trying to paddle toward the whales, and she was trying to paddle backwards away from the whales, and we went nowhere. So that's a good example of trying to make a joint decision when we don't agree.
That's really funny. Ron, you want to bring up anything else?
No, I think you covered a lot of territory in your questions, and it was fun to talk about these things. And I think that what you're doing in SITC, in the organization, and introducing scientists to each other and sharing results and encouraging young people to join the field is terrific. And I really salute you in what you're doing.
Well, thanks so much. It's such a pleasure to have Dr. Ron Levy, an icon in the field of tumor immunology from Stanford University, here with us today. Thanks again, Ron, for being with us.